Welcome, everyone. My name is David Park. I'm the director of the University of Ottawa Brain and Mind Research Institute. How many of you were here yesterday? Just to show of hands. Okay, a large percentage, but many of you haven't, so I will introduce the Brain and Mind. Every year I do this, and every year it gets even more true because the way to solve our problems with brain disorders and understanding the brain is to work together. We here in Ottawa are incredibly organized, incredibly dedicated, amongst 5 academic hospitals with their research institutes and 6 or 7 faculties within the University of Ottawa, and let's not forget Carleton University. Who's one of our incredible partners. So I just want to start off by saying, and I'll be very, very brief, that most of you who have known me probably knows me as a bureaucrat. I'm up here talking about the brain and mind, shilling for it, but I am also a scientist. I'm a molecular neuroscientist, and my 2 great passions are stroke and Parkinson's disease. And those are my 2 babies, and if you had perhaps to choose, like perhaps some parents do, I only have 1 child, so I don't have to, but about the advantages and disadvantages, strengths and weaknesses of each one of your children, it's difficult, but if I had to choose in terms of stroke and Parkinson's, what has happened between the 2 in direct impact for patients in recent times, I have to say we've made some incredibly exciting advances in stroke, whether it be stroke prevention, whether it be acute stroke, and what we'll be talking about today is how we progress forward with stroke recovery. So, we have an exciting panel of speakers. We're really blessed to be able to put this together for you. You guys are very, very important, and so without further ado, I'm going to introduce your moderator today, who is Sophia Goken, and she is a nurse specialist, a stroke prevention nurse specialist. She works at the Ottawa Hospital. She's also an Ontarian, and she's also been doing this for 16 years. And is a proud member of the Canadian Stroke Consortium, which is the organization within the Brain and Mind that does all things stroke. So, Sophia, I hand it over to you. Thank you.
Thank you. It's a pleasure to be here with you tonight to moderate this event on the groundbreaking topic of stem cell research for stroke. We have often heard that the pessimist sees the glass half empty, and the optimist sees the glass half full. But the scientist sees the glass completely differently. The scientist sees the glass completely full, half with liquid and half with air. And in seeing things differently, innovations and new treatments emerge. And tonight we look forward to hearing from our panel of leading Canadian researchers more about what they see for the future of stem cell research in generating novel approaches for the treatment of stroke and stroke recovery. The theme of our night reflects on Mr. Hockey, as Gordie Howe, who suffered a stroke in 2014, received stem cell treatment in what was dubbed the miracle in Mexico. And so in the spirit of Mr. Hockey, we have a Gordie Howe hat trick lineup for you. The Gordie Howe hat trick was named for a game in which a hockey player achieved 1 goal, 1 assist, and 1 fight. And our panel is going to deliver on that. I'm going to provide you a brief overview of our panel and their topics for tonight. Dr. Dale Corbett is a professor of neuroscience in the Department of Cellular and Molecular Medicine at the University of Ottawa, and the scientific director and CEO of the Heart and Stroke Foundation Canadian Partnership for Stroke Recovery. He is going to walk us through the goals for stroke recovery and set the stage for stem cell research and how it might play a role in these therapies. Dr. Duncan Stewart is the CEO of the Ottawa Hospital Research Institute and the president of the Ontario Institute for Regenerative Medicine. He's going to share with us the ways in which stem cell research have been used to assist in other health conditions and share with us his experiences in traveling with W5 to visit the stem cell lab in Mexico, which treated Gordie Howe. And Dr. Dar Dowlatshahi is the Associate Professor in the Department of Medicine and Scientist at the Ottawa Hospital Research Institute. He's also a stroke neurologist at the Ottawa Hospital. And he's going to explain for us the vision for stem cell research in stroke and why this is one worth fighting for. And so I invite Dr. Dale Corbett to come speak with us. He's an expert in neuroplasticity who's dedicated to restoring lives through research. He's going to describe for us the current context and goals for stroke recovery from his perspective.
It's a pleasure to be here speaking to you tonight. And initially I had loaded up another presentation, actually a lecture I was giving this morning, which is on a completely different topic. And any of you would like to stay later, I'd be happy to talk to you about that. But I guess I better just focus on the topic tonight, which is stem cell therapy as an emerging treatment for stroke. Now, my perspective is one very much of a basic researcher, and I hope you'll appreciate that because stem cell therapy is such a new way of going forward, a lot of the research that's been done with stem cells has happened in preclinical animal models, because you can't do a lot of this fundamental research in people as of yet. So I'm going to give you that kind of perspective from the preclinical side and explain where we are, and hopefully it'll set the stage for our other speakers tonight. So a few facts about stroke. So we have about 62,000 strokes in Canada per year, and we have currently at least 405,000 people living with stroke and then disabilities. And what's particularly troubling is that our estimates indicate that the incidence of stroke will double in the next 2 decades. So by the year 2038, we expect those numbers to really be almost the entire population of the city of Ottawa. So just imagine if every single man, woman, and child in Ottawa was disabled because of stroke, just what impact that would have on the healthcare system. So this is a very expensive disorder because it's a chronic disorder, and it costs the healthcare system in direct and indirect costs of at least $3 billion per year. And while, because of the advances in acute stroke care, many, many more people are surviving today, and in fact, stroke has really changed from one of survivability to one of chronic disability. So many of these people who do survive long-term have chronic disabilities at 6 months. So they've recovered from the initial stroke, but they're still impaired. They're maybe not able to carry out their activities of daily living. The other thing that's begun to emerge is that many patients, after making the initial recovery or functional gains after stroke, they don't just stay at that level. Many people actually progressively get worse. And there are reasons for this. So we're really starting to think that not only is stroke a chronic disorder, but in many individuals it's also progressive as well. And this means greater impairments and more demands on care. So I want to share with you some data here. On the left we have some graphs which show the recovery of upper limb function in people who've had a stroke over time. And I don't know if you can see this very well, particularly at the back. I can barely see it from here. But what it really shows is that people in the first weeks after stroke begin to recover quite quickly, and then it reaches a plateau. These curves level off and plateau. And at that point, they really don't get much better. If they do get better, it's usually because they develop compensatory techniques. So what I want you to appreciate is a lot of that rapid recovery happens in the first few weeks, and then it really slows, and you might say even stalls or plateaus. Now if you look at this graph, this is the same kind of recovery profile, except that it's in a rat who's had a stroke, very similar to what these people have had. And we're measuring their upper limb function, and they reach for these yummy little food pellets, which they like very much. And you see basically the same kind of profile, initial rapid gains in recovery, and then a plateau. And then no matter really what we do with these animals, once they've plateaued, they don't really seem to recover any more than that. And that's pretty much the same case that we see in patients. So the whole thing about stem cell therapy is that, as was used on Gordie Howe, is that the hope is that you could change these recovery profiles. You could have people recover more quickly and more completely. So instead of plateauing here, maybe with stem cell therapy you could plateau here, closer to a normal level. So that's the real goal. Now, is it all hype, or is it really something that might be achievable? So that's what we're kind of here tonight to talk about. So stem cell transplants have come a long ways, and Dr. Stewart will talk much more about this because he's really a stem cell expert. But some of the early challenges with stem cell therapy was that they could be rejected, because you might be taking a foreign cell, putting it into a patient, and then the immune response would be such that these would be rejected. Or you might not get good survival. You put in like millions and millions of these cells, and then a short time later you find that almost all of them have died. So how are they going to do anything if they're dying off so quickly? Well, a lot of these problems have been solved now with different growing cells and harvesting them from different sorts of tissues. So, for example, instead of embryonic tissue, which was quite controversial because of the ethical issues involved, now it's possible to get cells and take them from other tissues like an umbilical cord or from bone marrow, which is quite common, or skin, or even from teeth. And then what you can do is you can change the genetic complexity of these cells and turn them into practically any kind of cell you want in the body. Could be something like a brain cell or some other type of cell. And then you can grow them, expand them, and then deliver them to patients, either intravenously or in some cases directly into the brain. So the main thing is that there are many, many sources of cells now, and we figured out ways to get around many of the early problems with rejection and survival and so on. So we're not really sure how they work, because once you transplant them or inject them into the person, they can do a variety of things. They can increase blood vessel formation, which can be very beneficial if you're trying to recover from a stroke because you need energy and oxygen. Can help with cell survival. So if you do this fairly soon after a stroke, it's possible that some brain cells may not die, that they may actually survive, so you would have a better outcome. And then I think the thing that really probably represents the greatest potential of stem cells is that it's a way to reorganize and rewire the brain. And that happens a bit anyway after injury. So if you damage the brain, it'll induce a whole host of neuroplastic changes, including sprouting of new connections and so on. But you can boost this response, perhaps, with stem cells. And we also have resident stem cells in the brain in a couple of places. And so these stem cells may also, given from outside the body, may mobilize the ones that reside within the brain and help them repair the brain. So a lot of the evidence, as I mentioned, comes from animal studies, and there have been a lot of studies now that have been done using different types of stem cells and then inducing stroke and looking how these stem cells can improve recovery. So one recent study concluded that a particular type of cell, which we'll be talking quite a bit about tonight, is mesenchymal stromal cells, or MSCs, have been quite effective in promoting recovery in the animal models. And we see these effects across species, which is really important. So it's been shown in mouse models and rat models, and even in some cases of higher non-human primates or monkeys in some cases. Many different cell sources and given in different ways. So they've been given intravenously, they've been injected directly into the brain, they've been given in through the arteries and so on, and it doesn't seem to matter. They all seem to have benefit. And many of the really higher quality MSC preclinical studies were associated with greater behavioral recovery. So that's a good sign, because what you don't want to see are quick and dirty studies having the best effects, because those may not be reproducible, but instead the pattern seems to be that the very best, most carefully conducted studies have shown the greatest benefit. So that's encouraging. So what might they be doing? So if we look at this graph and figure here, on the left, at the top left, you see a brain, and there's a darker area where the stroke has occurred. And what you're trying to do is to replace the function of that damaged tissue. Now, you might think that these stem cells might just go there and repopulate that area, and basically you've grown yourself a new brain. But that's pretty highly unlikely, and maybe that'll happen, but I wouldn't bet the family farm on that taking place. What does happen is that a lot of these cells will home in on the damaged area and infiltrate brain tissue around the stroke. And then what happens is a lot of what we call tissue remodeling. So we get formation of new blood vessels or angiogenesis. The insulation on nerve fibers, like the little plastic wire that's on electrical wire, that can be damaged. And so these stem cells can remyelinate or rewire these connections and improve synaptic transmission. Also, these stem cells are a source of neurotrophic factors. Neurotrophic factors are made up of a lot of growth factors, and you can think of them as fertilizer for the brain. So if you want to grow new connections in your brain, early in life we have a high level of these growth factors. So after injury, if you really want to grow new connections and try to restore function in that way, a source of neurotrophic factors is important. And one of the things that these stem cells represent is a source of growth factors. They can also alter the inflammatory response. We know that an inflammation is important for healing, but also in many cases it can contribute to secondary damage in the brain, which could impair function. So the thing to take away from this slide is that these MSC cells can do a whole host of things. So they're kind of like a cocktail. You know, they're sort of like a poly pill, if you like. They're not doing just one beneficial thing, but potentially at least 5 or 6 and probably many more beneficial things. So right now we have more than 40 completed or ongoing trials clinical trials using the variety of stem cells. The intravenous route is by far the most common, and there are some studies that are delivering in people the stem cells directly to the brain, and I'll talk about one of those studies in a moment. Most of the trials are delivering cells in what we call the acute phase, so that would be 1 to 7 days post-stroke. So what's happening then is the injury, to some extent, is still evolving. So by giving stem cells, then you have a chance maybe of limiting some of the secondary damage and particularly reducing things like inflammation. Later on, when you get into the early subacute or the chronic phase, which happens way out in time, that may be a more challenging time to induce recovery. Now remember from my earliest slides, when patients and animals plateau, it's in that chronic phase. And right now, there's nothing much that we can do to improve recovery at that point. So one real question is, can stem cells do something in either animals or in people whose recovery has plateaued? The other thing about these clinical trials that's very important to keep in mind and to think critically about them as opposed to just taking everything at face value in the media is that many of these trials are small. They don't really have enough subjects to really come to strong conclusions. And if you think about the Gordie Howe story, it was an N of one person. So it's very difficult to really know from that, to draw strong conclusions, and we'll hear more about that. So many of these trials are really just testing safety and feasibility. So these are some of the data from the animal models, the most recent kind of findings. So one thing is that there's very much a pro-inflammatory state that happens early after stroke. And these inflammatory cells come from the brain, and they also come from other sources in the body. And one thing that has emerged in very recent animal studies is that the intravenous MSC cells, given 24 hours after stroke, can really normalize and minimize this inflammatory response. So this could have enormous benefit early on if you give these cells. And there's been a recent clinical trial where they've just started to analyze the data. And in this trial, it indicates that these same MSC cells that are given to the animals, given up to 36 hours post-stroke, can improve clinical outcome. So that's encouraging that this relatively early treatment with this type of cell can be beneficial. But I've been particularly interested in my own research about people whose recovery has plateaued, who got rehabilitation and it didn't work very well, or never got TPA or some other treatment, and they're kind of stuck. And very often I hear from people like this who say, is there anything that can be done for me, because frankly, I don't like the state I'm in and I would really like to get better. Can you do anything with those people? So there was a study published last year in the journal Stroke by Gary Steinberg's group, who's a clinical neurologist at Stanford. And they took 18 patients and implanted them with these type of cells that I've been talking about. And what was really interesting in these patients, and these are MRI scans of a patient, 39-year-old female who had a transplant and then followed along in time, is that all of these patients, unlike the other ones I was talking about, these were all in the chronic phase. So they had recovered as much as they could and then plateaued at least 6 months, and many of them were several years after their stroke. So they delivered these stem cells and then measured various types of clinical outcome measures, which are quite good ones. And the one that's really best is something called the Fugl-Meyer score, which is a very sensitive measure of, among other things, upper limb impairment. And they saw an 11-point change in the Fugl-Meyer score. And these are in people who had plateaued and weren't getting any better. And so this was very, very encouraging for a number of reasons, because one thing that you might think about is that maybe after a few weeks or a month, all the plasticity in the brain is basically used up and you can't re-engage it in any way. This suggests that that's not true, that implanting these stem cells late, even when things have plateaued, you can maybe reopen that plasticity window, at least to some extent, and these people can make some functional gains. So that's really, really encouraging. These patients have been followed out, they're doing well. A number of them have maintained the functional gains that we're seeing. Some of them were quite dramatic, and again, small ends, they don't really have proper controls and so on. But some of them, you know, one lady in particular was in a wheelchair and now she can jog, albeit maybe not quickly, but she can jog, which is quite a remarkable transformation. So what might be happening is these stem cells are altering factors out in time that limit recovery. We know from very basic research that there are a number of genes and proteins that begin to get expressed 30 days or more after a stroke that in a way put the brakes on recovery. So initially they're not expressed, but as you go out further in time from the stroke, you can kind of think of these brakes coming on. It's kind of like trying to drive a car and you're pressing on the gas pedal and the other foot's on the brake, and you're not really getting anywhere. So maybe that's what's happening with stroke recovery. So if you could somehow get the brakes to come off a little bit, you could go forward and recover more. And so one of the things that these stem cells seem to be doing is removing some of these brakes that limit plasticity. So again, this is new research showing that these stem cells are changing parts of the kind of brain matrix called perineuronal nets. And we'll have a test on this later on for you, so I'll be asking you all about to redraw these things for me. But all you really need to know is these perineuronal nets are not present early in the brain when the brain is rapidly growing and new connections are forming. And then as we get older, these perineuronal nets start to form, and they really limit plasticity in the brain. But what's interesting is when the brain gets injured, they open up a little bit and that plasticity reemerges, almost like a juvenile type of plasticity. So one of the things that the stem cells seem to be doing is kind of disrupting these perineuronal nets and maybe allowing some plasticity to happen that otherwise would be slowed. So I think the future is really looking at multiple targets, giving stem cells early after the stroke to target maybe the inflammatory response, and then later on, maybe you're going after something else, these perineuronal nets or other factors that limit recovery and prevent us from getting more full recovery. So I think they're really producing their effects through multiple mechanisms. They appear safe, and we'll hear more about that from our other two speakers tonight. But we still need to answer a lot of key questions about what are the best cells to use. Are these MSC cells the best cell? Probably not, but you have to start somewhere. And they may be helpful, but we may progress to other types of cells or multiple cells. But I think simply doing rehabilitation for stroke patients will not get us to the next level. What we're doing today really isn't greatly changing the life of patients from 20 years ago. It's still pretty much the same place. So if we want to get a game-changing change in the recovery, it's going to be coming through interventions that I think like these to augment plasticity. So the Canadian Partnership for Stroke Recovery, in partnership with the Ottawa Hospital Research Institute and the University of Ottawa Brain and Mind Institute, hopes to initiate the first stem cell trial for stroke in Canada in the next couple of years. And now you're going to hear more about these attempts from our next two speakers. So, thank you.
So I was going to give you a very brief refresher on stem cells. You heard that from Dale Corbett, but since he's going to be giving you an examination, I thought we could benefit from just going over the high points again. So you all, maybe you can tell me what a stem cell is. Well, a stem cell is a cell that has two important functions. One, it can replicate itself. That's very important because you have to amplify. So you only have so many stem cells, but this is a cell that can continuously replicate without stop. So that's important. And the other important characteristic is it can replicate in a way that's asymmetrical. So it produces another stem cell, but also what's called a progenitor cell. So this is a more committed cell that will go on to produce a brain cell, a heart cell, or something else depending on the environment and the cues that it sees. Is this, my sound is strange. And then, of course, that progenitor cell can go on to produce any cell in the body if it's a pluripotent stem cell, or if it's a tissue stem cell, more restricted, so just certain cells in that particular organ, like a brain or a heart. So where do we get stem cells from? Well, this is a fertilized egg, which gives rise to a blastocyst, and that inner mass of that blastocyst, very few days after the beginning of the development of the embryo, contains cells that are called embryonic stem cells. So this is the cells you heard of. These are the real super stem cells. They can do absolutely everything. In fact, that's how we come about. They make us, right? So they've got few cells in the blastocyst. All the cells in the body, all the organs and tissues are produced. And they do that again by producing a whole variety of these progenitor cells, and then finally all the tissues in the body. So you might say these cells are the most powerful cells. Why wouldn't we use them? Well, they do have advantages. There's an endless source of stem cells because they can divide without limit, and they do produce all the cells in the body. But there's a big but. Only very few clinical trials have been done, and that's because of concerns about safety. Because they can replicate without stop, they can also produce unwanted tissue, even tumors, even neoplasias. Also, there's concerns about rejection, because necessarily these cells wouldn't be autologous, wouldn't be your own cells, they'd be coming from another individual, and then there's also the ethical concerns, and that's really been a big problem. However, fortunately, we can get stem cells from other places. So all of our tissues, all of our organs have stem cells, and as Dale Corbett told us, you can get stem cells from these organs and tissues. And this is just a summary, but really anywhere in the body there is stem cells within those tissues. And the stem cells we use very often come from the bone marrow or from the blood. And they seem to have quite a lot of interesting properties that are useful in trying to repair the brain after a stroke, for example. Now I'm going to put a little advertisement in. We're not new to the block here. Ottawa is a powerhouse when it comes to stem cell therapies. In fact, we're leading the country in terms of running trials using various kinds of stem cells to treat a number of very difficult problems. So the heart attack trial ongoing now, we're about halfway through that trial. This is genetically engineered stem cells to actually recover their activity in patients and their patients' own stem cells. So 37 patients have been enrolled. We're just starting a trial in a rare orphan disease called pulmonary arterial hypertension, and that'll be across Canada. 45 patients, doesn't sound like a lot of patients, but this is a 1 to 2 in a million disease. So you can imagine this is a lot of patients. This will take us 2 or 3 years and going across Canada to do. But this is very exciting. We're hoping to regrow blood vessels in the lung. A trial for septic shock called the KISS trial. We've done a phase 1 safety trial, so we've been able to show in 9 patients that this is well tolerated in patients in the ICU that have severe septic shock. And we're now starting a Canada-wide trial which has enough patients so we can test whether this is working. Do patients, and this again is anti-inflammatory, do patients recover better, have less organ damage, and live, and more likely to live with this treatment. And then you probably heard about multiple sclerosis. We're also nearly complete in a study using stem cells to try to help patients with multiple sclerosis, and we've completed enrollment at 20 patients. We produce all these cells right here at the Ottawa Hospital, just down the corridor. And this is one of our rooms, our cell production rooms. So we have a very sophisticated system with these biospherics isolator units where we can maintain very well-controlled, very clean environment for the cells. Everything is monitored. And the cells are harvested from the patient, for example, come into these isolators, and all of the manipulation is done within this very controlled environment. And out at the end comes the final product that we can deliver to our patients. So we have a lot of experience in doing this. So what about the brain? Well, as you heard, we're very interested in launching an innovative trial to see if we can improve the repair of the brain. Meanwhile, there's a lot of other places you can get stem cells. So it used to be stem cell tourism in countries in the Caribbean and elsewhere. Now actually it's all over North America, all over Canada, that these places are popping up. Stem bucks, over 1 billion cells. And there's a problem with this because Very often the stem cell, this has not been regulated, for example. The cell products are not well characterized. They may not even be safe. And no data is being collected. It's like McDonald's or like Starbucks. This is a commercial enterprise to part you from your money, and there's no guarantee that you'll get any benefit. In fact, there's risk that you'll get harm. So this brings us to Gordie Howe, and hopefully this will play. It's a 1-minute video that Avris Favreau provided for me. Can we, sorry, let's pause that. Can we have the sound? Okay. Okay, put that in there. Oh yeah. So we'll try that again. It's not working. It worked when we tested it. Israel, can you help me?
While you're setting us up,
Yeah, we listened for it. I know why that is.
Damage and remarkable recovery.
That's awesome.
And hockey legend Gordie Howe's battle to win back his life.
Gordie, that's fabulous.
We missed the whole thing, so let's try this again. Now with everything working. Okay. There we go.
They call him Mr. Hockey. A legendary player now facing the toughest fight of his life.
We didn't think he'd last a month.
Struggling back from a crippling stroke.
Couldn't talk, couldn't communicate.
With the help of a groundbreaking new therapy.
There was just something in there that was not going away.
Gordie Howe's family decided to bring him here to Tijuana, Mexico.
Avis Favreau.
What shape was he in when he arrived?
Couldn't speak at all.
With a new and radical medical treatment.
We need to step up to the plate and find ways of doing that here in Canada.
The devastating damage and remarkable recovery.
That's awesome.
And hockey legend Gordie Howe's battle to win back his life.
Gordie, that's fabulous.
Okay, so as you know, Gordie Howe suffered a very severe stroke actually around Thanksgiving in 2014, and then his family received a call from Dr. Maynard Howe, who's one of his sons, and a guy called Dave McEwen, who was CEO of this company, StemMedica. Now actually, this David had been with the Detroit Red Wings in some capacity, so there's a connection there, and that's how that call came to be. And they were told that they had an experimental stem cell treatment that could potentially help Mr. Howe. So just before Christmas, he was taken to the Santa Clarita Clinic in Tijuana, run by a company called Novastem. And he received 2 cell products, not just 1, but 2. He received fetal neuronal progenitor cells. These are neuronal stem cells derived from a fetus. And they were injected into his spinal canal one evening, and the following day he received 90 million of these MSCs, mesenchymal stem cells, that you've heard about, and these were given intravenously. And one day after this treatment, he had a remarkable improvement, and it's reported that he got up and was beginning to walk even one day after. So I was part of a team with CTV, Avis Favreau, that went down for W5 to look into this miracle and see what was going on. And first we visited San Diego, and StemMedica is a company that makes the cells. And then we traveled to Tijuana to see the clinic where these cells were delivered. So first of all, you know Tijuana is quite close to San Diego, so it's only about 20 miles. So if you drive at the speed limit, it's 34 minutes, but our car at 4 in the morning went much faster than that. And there was no wait at the border. The other thing that I learned is it's absolute day and night between San Diego, California, and Tijuana. This is the wall, the border between between California and Tijuana, and you see immediately, and this is not the nicest part of San Diego, but there are places where you have these beautiful suburbs, and then right on the other side you see sprawling urban, sort of really ghetto, just completely different worlds. So this is the clinic, Santa Clarita Clinic, and we were taking our tour, and one of the first things I saw was where they actually prepare the cells for injection. The StemMedica makes the cells, they're frozen, they're sent down to the clinic, and then they're prepared in a special facility, and they have very good standard operating procedures, so I was quite impressed really with the way that they did that. I didn't know what to expect to begin with. And then this is, I was watching one of the delivery procedures occurring, so this is those neural fetal stem cells being injected into this gentleman's spinal canal through a lumbar puncture, and there's the anesthetist putting in the needle and injecting the cells. So, did the stem cells help Mr. Howe? Well, Mr. Howe was examined by a prominent neurologist in the San Diego area called Dr. Kramer, who's head of neurology at one of the major hospitals. And he was actually quite impressed by the recovery. So he said that this was a remarkable degree of motor improvement for anybody with that severe stroke. But unfortunately, this improvement did not last. So Gordie Howe underwent a second cell treatment in the summer of 2015, so about 6 months after his initial treatment. And as you know, last year, he unfortunately passed away. So as Dale had said, in the absence, this is the N of 1, we have one patient in this study. So in the absence of a carefully thought out and performed clinical trial, we will never know for sure whether these stem cells had any real benefit for Mr. Howe. So I did a report card on what I saw, and this is the results. So this is the StemMedica on the top, and actually I was very impressed. I went through their facilities, saw how they made the cells, how they characterized their cells, and they did this very well. So this is one of the best facilities I've seen in terms of the quality control for cell manufacturing and the characterization. So I gave them an A+ for both their bone marrow cells, the MSCs, as well as their neurostem cells. So they put a lot of effort into that, and that was impressive. Also, I was quite impressed with the procedures that were there for cell preparations, and I showed you that room at the Santa Clarita Clinic, so that was good. In terms of the delivery, I was not so impressed. There were no real protocols for doing this. As you could see, it looked like being done in a very casual fashion. In fact, one of the new nurses there that had been working there for a month or two came to me to tell me how she enjoyed working there. In fact, she had had back pain, and that same anesthetist had helped her by injecting stem cells into her back. There was no consent, there was no study, there's no regulation. You can do what you want. It's really the Wild West. So that's a bit of a concern. In terms of the study, because they claim they're doing a study, and they actually showed me a protocol. And basically that protocol, you could put in, it was fill in the blanks, right? So you're giving stem cells for blank, whatever disease, you put in the disease, and the number of cells and all this. It was not a protocol at all. And so I gave them a D- on their clinical trial protocol. Informed consent process, again, was nowhere near. There really was a non-existent, so an F there. And they don't collect any data. So I asked about Mr. Howe and other people that had received similar stem cell injections and what were they doing in terms of follow-up tests. They say, well, we don't do any visits, we don't do any tests, but we ask where they come from to send us results if they do anything. And I asked, did they get any results? And they said, well, no, they don't send them. So they're not collecting any data in any systematic fashion whatsoever. So again, unfortunately, we will learn nothing. Now, I don't know if Mr. Howe paid any money. I think not. I think this was provided free of charge because of whom he was. But certainly everyone else that comes to this clinic pays money. This is a business. And that's how they afford this nice clinic, is because they charge significant amounts of money for people to come and get stem cells. So what is it that we need to do? So I think we need to really understand what's going on. We heard some beautiful work from Dale Corbett. We need to do animal studies to really look at the effectiveness of the cell therapies. Just to tell you, there's no animal studies whatsoever to show effectiveness of the StemMedica cells. They produce beautiful cells. They've done no studies of how well they work. And moreover, they use two different kinds of cells, and they've not done any studies to show that you need to use two different cell types, and they have no studies that show there's any benefit to combine the MSCs with the neuroprogenitors. It sounds like a good idea, but they have no support for it. And again, you need to design a very good clinical trial to assess both the safety, very, very important, as well as the efficacy, that is whether or not the treatment works. And that's something we have great experience with here in Ottawa, at the Ottawa Hospital and the university, and you'll hear that, I think, in spades from the next speaker. So again, just to go back to Mr. Howe, there was no attempt again to collect any data, so we will learn nothing from this. So I will end there and ask the next speaker to introduce. Thank you.
So now we'll have our third speaker in the Gordie Howe hat trick, Dr. Dar Dowlatshahi, to speak with us more about specifically how this can be used to treat patients in stroke. Dr. Dowlatshahi is a stroke neurologist and stroke researcher.
So my day job is a stroke neurologist. I take care of patients in the acute setting and in the prevention clinic where I work closely with Sophia. But I also do stroke research, and one of the areas that we're just starting to look at is stem cells in stroke. Now, I don't have much experience or any experience in stem cells. I have the experience in doing the stroke research. So this is where we formed a natural partnership with Dr. Stewart and Dr. Corbett, bringing the recovery expertise and the fantastic stem cell work that's being done in other diseases. It just seemed like a natural extension to bring that expertise into stroke and try and help out both local stroke survivors and, of course, the stroke field internationally. So the big question is, where are we at right now? Well, here's what we don't know. We don't know what type of stroke, first of all, can be helped with stem cells. There's many types of strokes. There's stroke survivors in the audience right now, and I'll guarantee you that you can randomly pick two people who are stroke survivors here, and their strokes are in no way similar. May share some features. There may be some difficulty with language function, perhaps some difficulty with movement, but in terms of the type of stroke they had, the part of the brain that was affected, whether even if it's a bleeding stroke or a clotting stroke, there will be differences. Stroke is not one disease, it's 100 diseases, and this is a problem when we say, let's give stem cells to stroke. What stroke are we talking about? What type of stem cells? We just heard there's tons of types of stem cells. Most people don't even know that. They hear stem cells and they immediately think either of the old embryonic debates, which no longer applies because we don't use those, or they just think of the new stem cells and sure, let's just give it and see what happens. And as you can see, that's being done in North America now. Do you go IV? Do people even know that's an option? You can give stem cells into the blood, or do you need to put them directly into the brain, like that recent Steinberg study that Dr. Corbett showed us? What about the spinal cord? We just saw that being an option as well. A bit of an atypical option, I have to say, but it's a possibility. When do you start? So stroke is an event, and you have a recovery process after the event. Do you do it right when the event occurred? Do you wait until the person has a chance to recover and then plateau? Do you wait 5 years? Do you wait for the stroke survivor who has done 10 years and now says, I want more? When? It's all different. Our stroke survivors who've been through this for 5, 10 years will sit down with a person who's had a stroke in 3 months and tell them, things are going to change. This is not you at 3 months, you're going to be different at 1 year. Well then, should we be doing the same treatment for the person at 3 months, 1 year, and 10 years? What dose? We don't even know where to begin with that one. How many doses? Gordie Howe got 2 6 months apart and then passed away, unfortunately. So we actually don't know anything about stem cell research for stroke. We don't, and that's the truth. Yet, you can get some right now. You can hop on a plane and go get some. That should concern you, and that concerns a lot of people. So it's important to have hope and optimism, but I think what we need is enlightened optimism and cautious hope. Okay? These are not doctors trying to tell you not to do things to your own body. These are doctors that are telling you if the doctors don't know what's going on, how could somebody be selling you this product and telling you they know what's going on? I want to highlight one of these things that are said there, no negative side effects. Just keep that one in mind. That's a real ad, by the way. So has there been much research? Yeah, there's been quite a bit. And in fact, there's something called a systematic review where you take a bunch of what you consider to be reasonably good studies and put them together. And this was done between 2015 or 2005, 2014. All these studies were put together. There's been many since then, but these were one that brought together 481 patients to try and get a sense of what's going on. And the bottom line is it showed that there was some benefit. It showed that there's a potential benefit of stem cells. So what's the problem if there are studies that are showing some benefit? When you look closer at what these studies were, only 9 of them had control groups. That's important because there's something called the placebo effect, where you didn't even get a treatment, but you get better. It's about 30 to 40% of the benefit of a product, a drug, a regimen, whatever, a food, an exercise, is the placebo effect. So you have to have a product or a treatment that is better than the placebo effect. No control group, and you can't do that. So I'll give you an example of this. There was a nice study, as Dr. Corbett showed, that had all those patients, 16 of them in total, that got the stem cell infusion, and they got better at 3 months. There was no control group. This could have all been placebo effect. And you know why I know that? Why the person in the wheelchair then gets up and goes jogging when they're followed up 6 months to a year later. Because I'm a stroke neurologist and I see stroke survivors. And I still remember a very devastating stroke in a 45-year-old, Mr. P, who I still see every year. And at 1 year, he was in a wheelchair, he was there with his wife, he was crying, he was pelagic on one side, couldn't move one side of his body. 1 year later, he has no cane and he's driving his own car and drove in to see me for follow-up. Was I surprised? I was like, pleased? Yes, I thought it was fantastic. I'd love to see my guys recover over time. Did he get stem cells? No, that was the natural recovery. Some will say he's gotten lucky because there's a lot of stroke survivors who did not get that level of benefit, but there are the rare ones that do just get better. So you need to show that it was the stem cells and not just the natural history of the stroke, and that's why you need a control group. 5 of the studies had greater than 20 patients. When you're dealing with 15, 16 patients, you get flukes and lucky things happening. And when you're dealing with not very nicely done research, such as these clinics that don't collect any data, you happen to show the ones that do well, like Gordie Howe, and you don't show the other 1,000 patients that didn't do so well. Where are all the other patients that came out of this clinic who's giving stem cells all the time? 2 studies that were published that were considered good turned out to use the same patients and the same data published in 2 different journals and claimed it was 2 different studies. This happens in the field, unfortunately. 6 studies didn't account for the type of stroke. Didn't even write down, was it a bleeding stroke, was it a clotting stroke? Did not account for it. There was a large stroke study that many of us were involved with in North America, which was started really well, ran out of money, and then left North America and went into another country. When the stroke study was done, it was negative. Afterwards, the original DSMB, the Data Safety Monitoring Board, analyzed the results and looked through it. They found numerous patients enrolled into the study who had never had a stroke. This happens. Okay? This happens when you do research in areas that are not very strictly controlled. There have been inconsistencies in all these studies about routes of administration. Even in the same study, one person gets an IV, one person gets it in the spinal cord and whatnot. How can you compare these things? You can't. The cell types are all different. Everything is different. We really don't know. One thing I did find, though, is there was a 13 to 15% chance of death, seizure, and infection through the variety of these processes. I'd just like to remind you of the ad that says no known negative effects. I do consider death to be a problem for treatment. Seizure and infection, I suppose, can be treated. So, you know, there is clinical improvement that's been suggested. I think there is hope, but the point is that it's not ready to go purchase yet. And unfortunately, when you're desperate and you're frustrated and you're not getting better and there's all these barriers ahead of you, sometimes it's that frustration that leads you to go have a leap of faith. But I would say that it's important for us in both the patient, the stroke survivor, the caregiver, and the medical community to practice the cautious optimism, okay, the enlightened optimism, to look at things. What does stroke do? Stroke naturally improves over time. This is a curve. You can just put recovery over there. The scores don't matter. And this is time. As Dr. Corbett showed, early in the stroke, that blue line is your average patient in an average hospital. gets better usually in the first 3 months. There's a bit of a spike. Some people have devastating strokes and it takes a few weeks before they start improving. Others get fantastic improvement over 2 weeks. You have probably all met stroke survivors that have had no symptoms whatsoever. You have no idea they're a stroke survivor unless they choose to tell you. Some people get very lucky, others not so much. But everybody does plateau. We have been able to show with specialized care, which I use that, and it actually really means physiotherapy, occupational therapy, and speech therapy. Okay? In a standardized unit called a stroke unit, you can make a difference and you can bump people up. But here's what we know is the average stroke pattern is towards improvement. If you're going to have a neurological disease, as horrible as stroke is, it is the only neurological disease that gets a little bit better every day forever. Okay? And one thing that we have learned in the last 3-4 years is that even at 2 and 3 years, there is a small amount of improvement every day. It's very small. And the patient, the stroke survivor, does not notice it, neither does their family members. But the person that notices it is the person that they see once a year. And a stroke survivor, and I know there's some nods in the audience because they know it when they go and visit family in another country or another city and whatnot, once a year, they go, my goodness, you look better. And they go, no, I feel the same. And you go, well, you had a cane last year. You don't even have a cane this year. Okay, that's a reality. And I don't get the benefit of seeing every one of my patients once a year. The rehabilitation doctors usually do that. But the ones that I do, because I do stroke prevention for some of them who have recurrent events and whatnot, I always document what I see when they come in so that I can show them the next year what I documented, because it's always better the next year, always. And usually the person's very frustrated. I remind them that they had a cane the last year. They don't have a cane this year. They weren't driving last year. You're driving this year, et cetera, et cetera. You were here with your family. This year you came by yourself because you can now do that. So people improve. When we give stem cells, we have to show that we are above this level of natural improvement. Go get stem cells today, you will be better next time, next year. Okay? Show that it's the stem cell. So our goal in clinical research is to deliver the therapy in a safe manner and prove that it works. There are ways to do this. There's a large group of people who have a vested interest in making people better using stem cells, and so what they want to do is set up a set of criteria, not rules really, but criteria to meet, to make sure that your study is rigorous and useful. Useful, not just to be used for a clinic to sell a product, but useful for the scientific community and for the patients and the stroke survivors. It's called Steps 3. We don't need to get into the technicality of it, but I'll try and break it down into some broad terms. The first thing they tell you to do is look at your selection of the stroke survivor. Do you go broad or narrow? Broad means we want everybody in, and that's what the clinics will do. Just bring everybody in, give them this, and it's going to work. But the problem with broad is it's harder to show a benefit because your diseases can be so different that this stem cell would have worked for this type of stroke and it doesn't work for that type of stroke. But by giving it to everybody, you can't tell that it's working. But when you're broad, you're more generalizable and you're more helpful. When you go very narrow, I'm only going to treat this type of stroke with this type of problem, you are more likely to show benefit, but less likely to help everybody. So you have to make a decision right at the beginning. Are you going broad or are you going narrow? Right now, I would argue, because we have no proof this stuff works, we need to start narrow. We need to really pick one area, show that it works, and then start broadening it. Works for this, does it work for that, does it work for that? But you can have the argument, what's the better way to go? What's your overall health? Well, if you pick the guy who's incredibly healthy to begin with, but had the misfortune of having a stroke, you're more likely to show benefit because there's not a lot of competing conditions like heart disease, lung disease, kidney disease that will cause side effects from your procedure or other problems. You can try work and fix the stroke and then have a heart attack. Right? So the healthy guy is more likely to succeed with these studies, but less likely to represent the person with the problem. Okay? I get young people who are otherwise healthy, who had a very rare thing happen that caused a stroke. They're not going to be representative of the average person who's 65 to 75 years old, has heart disease, smoked a lot but gave it up, started getting their health back together, and that's when they had the stroke, unfortunately. And after the stroke, they still are struggling with the heart and lung disease. That's a harder person to enroll into a trial and get successful results but that's the person that you're trying to treat in the community. So we have to make this decision when we make the trial. Where are we going to go on that spectrum? What about our time window? I've tried enrolling into what we call acute trials, right after you have your stroke. But turns out your recovery is really good at that period anyway, so it's very hard to show benefit. In fact, those of you who are stroke survivors may remember your first week. Scary, very scary. And then the last thing you want is some scientist like me coming up to you and saying, I'd like to give you an experimental treatment while we're here. I've tried that. It does not work well. Patients do not like it. Our patients tell us, you're scaring me. Okay, I'm trying to understand why my leg won't work and you want to put stem cells into me. We did a study like that. It was very hard to convince people to do it. What about the subacute, the weeks to months? Well, this is challenging because this is where we're trying to do our rehabilitation efforts and have already shown we can impact recovery through good rehabilitation. You really don't want to mess with something that is working. So some of us will argue that the chronic is where you want to go. The rehab is plateauing. People are done rehab. They're not getting better, or they're still getting better, but not as fast as they want. Maybe that's the zone to go for. But then you run into this problem of when you say greater than 3 months or greater than 6 months, that's a pretty broad time. The most common patient that'll call our office, I heard you guys are going to do a stem cell study, I want in, has had the disease for 10 years. That's very different than the person who's had the disease for 3 to 4 months and is just seeing what reintegration into community is like. Those are two very different diseases at that point, and we have to decide which one. You can't do both yet, not until we figure this out. And what's our endpoint? Well, do we go for a home run? That's called a global endpoint, make everything better. Okay, you had trouble walking, talking, thinking, concentrating, memories shot. Let's make it all better. What are the odds of doing that? Well, the clinics say they can do it, but I can tell you, it's pretty hard in any branch of medicine to get a global impact, especially when you're first starting a line of research. Or do you go domain specific? Like this gentleman here who had a language, specifically a language problem, but otherwise perfectly fine. And we can just say, we want to make your language better. Did it work or not? Okay. Again, comes back to that generalizability. We show that we can make language better, doesn't mean we can make your weakness better, but it's a start. And we got to start with something. There's other considerations. You have to look at what else are you doing with the stem cells. Are you also doing physiotherapy, occupational therapy? There was a study that suggested the stem cells don't work, but then combined with physio and occupational therapy, they do work, and it makes sense. You're putting something to regenerate the brain. It stands to reason you need to activate the brain to make that regeneration take. So we don't know these things. We're still trying to figure these things out. So we have to look at that. What about brain imaging? What if we don't see an improvement in the person's performance, but the brain actually looks like it's healing? It might just mean that we're measuring the wrong things. So maybe we should incorporate imaging. There's so many other things in that STEPS criteria and in our research that we have to consider when we design it. So when people will say, why is it taking so long? Why is it so expensive? Why can't it start now? Because of all of these things. It takes a long time to get it right. So what do we need? Well, I think clearly what we need is people to start adhering to the same criteria, if only so we can compare notes, so we can compare results, compare research, and not duplicate efforts, and not duplicate failures. We want to get successes that can be added together and figure out what's going faster. We have funding organizations, fortunately, in Canada and the US that are very strict with methodology, meaning wanting to make sure you're doing it properly before they pay you money. We want them to continue being that way. We cannot afford to waste tax dollars and hard collected money for research on badly done research. It has to be responsible research. In Canada, we have many organizations, including Dr. Corbett's CPSR, Center for the Canadian Partnership for Stroke Recovery, that is doing this, that is making sure the research is done properly. But I gotta say, a big problem is clinicians. We need our clinicians to be ethical. We need our clinicians to offer therapies that they know are safe and effective and not give false hope to our stroke survivors and say, of course we can make this better. And you want to watch out, because just because you're smart and you went through medical school does not mean you're not a snake oil salesman. Okay? Be careful with clinicians. Okay? In Canada, fortunately, for the most part, I haven't seen too much, but as Dr. Stewart said, things are starting to pop up. So what are we doing? We're very early. Now, when I say early, we've been working on this for over a year, actually. And we are still in the process of securing money. And we have secured some funding from a variety of organizations, including the Canadian Partnership for Stroke Recovery, including the University of Ottawa Brain and Mind Institute, which has hosted this event for you right now. They're actually a major contributor to this concept, and now we're going to the governmental agencies to try and get money to do our own stem cell trial because we have the expertise and the timing. This is where we are now. We did decide to go narrow. Okay, some of our stroke survivors won't be too happy about this if the problem isn't the one that they have, but we went with one that our stroke survivors told us is important, their arm. We're going to focus on the arm. We know language is important, we know leg, we know walking, but at the time point that we're going at, our stroke survivors are extremely frustrated by the inability to use their arms, so that we're going to pick that one to start with, just to start with. Okay, we're going to pick 2 to 4 months. I'd love to take the person who's had it for 10 years, but for the reasons I discussed, it's very difficult to take all these different time points. We're going to pick the one where we know there's frustration because recovery was really good. People went to rehab, they finished rehab, and they're not where they want to be. Because we can approach those people, we can identify those people, they're in the rehab center, and they're leaving the rehab center, feeding back to the rehab doc, "Doc, I'm not good enough. My arm is not good enough." Maybe we can change that plateauing we see of recovery and spike it back up again. It's something that we can try and go after. That's why we're picking this time point. We're going to go with a blood infusion. Our patients are very clear, so are our doctors. We don't like sticking things in people's brains. It makes no sense. You injure brains by sticking things in them. There's a higher risk associated with it. We have data from the cardiac work that Dr. Stewart is doing that shows injecting into the arm works on the heart. It stands to reason it's going to work on the brain. There are studies to show that because there's trafficking of the cells. There's things going on in the body that we don't understand. We don't think we need to go directly into the brain. And we're going to do something that's kind of complicated. It's called a crossover design. So one thing that our patients, so we went to stroke survivors and asked how should we do the trial when we do the trial. This was done over the last year. And one thing they all said is, look, we want to be part of the research, we want stem cells, but we'd really like to get them. We get it, you need a placebo group. We had a couple of patients who actually said, we get it, you need a placebo group. I just hope I'm not in the placebo group. There's a way around that. It's called a crossover, where what happens is you could be in the placebo group the first time. You get an injection around 3 months. Half of you are in the placebo group, half of you are in the stem cell group. You don't know it, we don't know it. It's blinded, it's called. And then we go through the study and see how you do for the next 3 months. And then we wait and cross you over. The ones who are in placebo come back for a second treatment, this time stem cells. The one where stem cells come back get placebo. We get to do 2 things there. 1, we do get our placebo control. 2, we get to see the ones who got stem cells if the effect actually lasts a whole year, because the second time they didn't get the stem cell. And 3, more importantly, all the stroke survivors get access to the therapy that they want when they enroll into the trial. It's a way of doing it. It's a very good methodology, actually, by one of our lead methodologists, Dr. Ferguson, who came up with that technique. And another little bonus in there that Dr. Corbett has been very adamant about. Dr. Corbett likes to exercise and tells me I should do more of it. He's very adamant about doing physiotherapy and exercise when you're doing these studies as part of it. So we do have some partners in our community who've actually been part of the brain and mind community for a long time, who are going to be providing us and the patients in the trial with additional physiotherapy above and beyond what they would normally get while they are getting the stem cell infusions for at least a 6-month period during the 1-year trial. So we are including a physical therapy and exercise component in addition. That's a bonus for the patients because they don't have to pay for it, but that's where we are. We're very hopeful. It will take us a while to raise the money. We are talking literally over $1 million a year to do something like this for as little as 15 to 18 patients. These things are incredibly expensive. We're working on it. Thank you.
Thanks so much to all our speakers. I'm going to invite the panel of presenters up here now and open the floor to all of you so that if you came with burning questions or if you have as many questions as Dr. Dowlatshahi has, you have an opportunity to ask them. We have a few microphones in the center, and if you need a microphone brought to you, I think some of them are mobile, is that right? Yes, do you want to receive the microphone or go ahead to
I don't think I need it.
Okay.
Can you hear me okay? I just have a very simple question. I'm not medical by any stretch of imagination, but how do the stem cells actually know where to go and how they work?
Maybe I'll start with that. So that's a $1 million question. If we had the answer, but we were getting some of the answer. So it depends. Stem cells can work in different ways. So when we began, when this field began, it was thought the stem cells would be building blocks to rebuild the brain or the heart, and they would actually turn into new heart cells or new brain cells, and that can happen, but it probably, with the cells we use and the way we deliver them, that probably doesn't happen very much. So what we've learned is they do a lot of other things. So they stimulate local mechanisms of repair. That includes, as you heard, regenerating the blood vessels, stimulating stem cells that reside in the brain to actually begin to repair better. And they also modulate the inflammation. So that can be locally as well. In the brain, we've heard that inflammation is bad and can induce damage to the brain. And so by modifying or reducing the inflammation, that can be beneficial. And that can occur not only in the brain, but in other parts of the body too, in some of the lymph system where the immune cells, the inflammatory cells come from. So they have actions which are, some of them are only local, and that would be rebuilding, becoming building blocks, and we know that's not probably very important in how these cells work. And they have other actions that can be both local and in the rest of the body to promote healing and reduce inflammation. But they do have ability to home, so they will go to areas of damage, so to some degree they will do that. But they don't stay there very long, so they stay there for days or weeks. But that's enough time to start this process usually. But much more work needs to be done on exactly what they're doing and where they are. And so that's something we want to try to address in these studies.
Maybe I'll just add a little bit to that. Early after the stroke, the tissue around the stroke will sort of send out chemical signals that will actually attract these cells. So we've done studies in animals where we've been able to label the cells with a fluorescent label so you can actually see them in the brain and watch them. So we'll inject it in one part of the brain and then let the animals survive a period of time, and you can see that they actually migrate in a certain pathway right to the area of damage. And it's really quite an interesting process. So that could be going on. You can't really easily see these migrating in the human brain, but it's likely the same kind of process.
We have a question in the center.
First of all, I want to thank all of you. As a stroke survivor for the past 18 years, and having worked with other stroke survivors and been the chairman of the board for the Stroke Survivors Association for more than 10 years, I have to say it is amazing to see the progress that has been done in the field. We talked a lot about physiotherapy and the work that hopefully will develop from stem cell research and physiotherapy combined. However, I wish to express our concern about the fact that only approximately 25 to 35% of all stroke patients receive physiotherapy. And how is that going to be overcome when you're doing your clinical assessment? Also, you have been talking, and I greatly appreciate that, about the physical aspect of stroke. What we were concerned about in the Stroke Survivors Association was the community reintegration. How do stroke survivors and how will stroke survivors reintegrate into the community? How will they deal with issues such as economic, mental, emotional, and other types of issues of this type, even after you supply the cure? That looks very exciting.
Dr. Stewart? No. Thanks, Abe. Abe, you raised a really good point. For those who don't know, about roughly 1 in 3 patients with stroke will have post-stroke depression, not just because they're depressed about the physical and cognitive effects of the stroke. These are people who fully recovered from stroke will actually get the depression, and in a lot of people this is under-recognized and leads to difficulties with reintegration. Upwards of 30 to 50% will have cognitive issues that are not easily evident, but clearly evident to the survivor and to the family members around them. There's post-stroke fatigue, which is very common, 30 to 50%. All of these factors are above the physical, language, and the other visible, if you will, effects that stroke has. All of these are barriers. And then even beyond all those barriers, as Ava has pointed out, there's the economic side of it. There's the fear. There's the anxiety that comes from it. There's the, I think we're a grown-up audience, so I could tell you I've had people that come a year later and ask me if they can start having sex yet for fear of causing a stroke. Right? So the things, there's so many aspects to this, and again, I would love to give you the home run. We would love to do that, but you can't. So what we can do on the research side is try and move things incrementally forward and try and make things a little bit better than they were before. So that's our first goal. That's number 1. Number 2, absolutely, there's a problem with the physiotherapy out there, and this unfortunately is not something that is an academic issue. This is something that's a government/resource issue. As you know, we're in a provincial type of healthcare system where resources are allocated, shared between conditions, locations, people, etc., to some formula that none of us really have access to, unless if you're part of it. And there's a finite pot, and someone's made the decision that this is how much there is for stroke, which maybe because if you put more heart disease or cancer doesn't have as much, I don't know. But there is a problem. So the way that, to answer your question, Abe, how are we going to account for that? Like I said, we have to start with a narrow patient selection. So what we're selecting are those who went to a rehabilitation facility, received the same amount of rehab, specifically, if you want to know the exact details, to either the Elizabeth Briere for 4 to 8 weeks, whatever it is that they got there, or Pembroke, because we also want to go out a little bit. This is not just an Ottawa thing. We want to go where we can. Those are the 2 centers that right now have agreed and are interested. So that's 1. So we ensure that there is a baseline level of physio that's occurred. And then 2, we are going to give them physio for exactly that reason. We have to. And I recognize that even if this is successful, what it's saying is if you are lucky enough to get into rehab, okay, which is 24% of people, I believe, okay, if you are lucky enough to have come out and been eligible for a clinical trial, which means your health was good enough that you could take the procedure, and then lucky enough to have 6 months of paid private physiotherapy and stem cells, you'll do okay. That in no way fixes our average stroke survivor, absolutely. It's the first step, is what it is, and then we use that for the next step and so on, and then we get guys like you to advocate.
There was a question here in front. I don't know if you want me to bring you a microphone.
Well, I don't know. Can you hear me? I'm just wondering about how narrow you can actually be. If you're only working on an arm, you mean the stroke is a little teeny part of the motor cortex, or these are very small strokes.
Not necessarily. I mean, we're not suggesting that you can't be in if you have more than an arm discomfort or so we'll take anything, but we do want a person with arm because what you call outcome measurement, so the way that we're going to decide whether there's an improvement or not is focused on that arm. So you could still have leg problem or language problem as well.
Yeah, I think we're all well aware that there are other issues like, cognition and depression that are extremely important to people, but we understand the motor system the best, and so when you're trying to work things out, it's where the light's brightest, so it's easy to work there relative to some of these other functional domains, and the outcome measures are very good ones, where, with cognition, and you talk to people about, well, how do we assess cognition? And, you get like 30 different answers. People don't know, but people all agree in how you assess upper limb function properly. So we start there, and then hopefully what we learn and some of those principles will be, we can extrapolate to these other functional domains.
There's a question back here.
Actually, there's 3 quick questions. One, I was surprised at the apparent number of non-scientific studies. 2, is there no overall body who can deny such a study before it wastes resources? And 3, what about an overall body dictating proper scientific procedures, or you don't get support?
Maybe I'll start with a general comment, and then Dar can probably talk about stroke. Many of these studies are done in different jurisdictions. So in Canada, in North America, Canada, and U.S., you can't do a study like this without getting a whole level of permission. So you'd have to go to the government, in our case, Health Canada, and you have to sit down. They'd be very interested in knowing about the safety aspects, but also trial design, all of these things. You have to fill out the same kind of paperwork that if you were a big pharma company wanting to do a trial, same thing. It's called a clinical trials application. It's a tremendously onerous thing to do. So fortunately, we have people that within the hospital that understand how to do this and help us do this. And then you have to, usually there's a face-to-face meeting, so you need to meet with them and go over all these details, and then you put all this documentation in your submission, and then they will look at it carefully and decide whether it's something they will allow to go forward. So that there is a lot of oversight. And then you have to get research ethics approval in every clinical site, every hospital where this is, or clinic where this is going to be done. And that again is a very rigorous process where a lot goes in. Again, the safety as well as the rigorous nature of what you're doing will be examined. But that's not true some places, right? I mentioned in Tijuana this young nurse that just had stem cells injected into her back for back pain. There was no regulation whatsoever. Again, the trials they're doing there's no government body that oversees that. It's really the Wild West. And there are many places in the world where that kind of thing can happen.
So we're saying the well-developed countries have the time, the energy, and the knowledge to keep it straight.
That's right. They learn for the obvious reasons to protect the public foremost, but also make sure that only the studies that are going forward, are ones that are worthwhile doing.
I think another thing that relates to what Dr. Stewart said was that these international organizations, like in the case of the stem cells, the STEPS initiative, they're really establishing guidelines for the rest of the world, all researchers to follow. So increasingly, it'll get more and more difficult to do studies and publish the results unless you absolutely follow these guidelines. So that will really help prevent a lot of this erroneous material getting into the scientific literature.
My question, just going back to that interesting scenario with STEMedica, there's clearly a huge commercial element in the United States, and I'm wondering, is there potential, or do you see other scenarios, for example, where STEMedica is producing these cells for legitimate studies in the United States or in Canada? Are you concerned about that element, that sort of lack of control that might happen if that commercial industry sort of starts to pop up here, or maybe it already exists, I have no idea? Or do you see it as a potential, something that could be worked with, or something that could be a benefit somehow?
So, STEMedica, as I said, they actually, I think, have produced a very good product. They put all of their money, all their resources into developing the processes for doing that, and validating and doing the quality control. So, what they haven't done is invested in the research to show that that product actually does what they think it does in appropriate animal models. But they are engaging in the U.S. in well-designed trials. So, and I wouldn't be concerned. The product they have, you know, if we were using the MSCs from Stemedica, I think that they're probably very high-quality MSCs, and that might be a good product to use. I would like to get the preclinical studies. I like to be able to squirt them into his rats and tell me they do work before I go. But you know, I think they're a high-quality product. The concern about, you know, there are many other outfits that are popping up, and in fact, there are. This is even happening in doctors' offices because there is. I think there's probably more than one, but there's devices that are basically like little washing machines you can have in your office. You can take a little bit of fat or a bit of bone marrow or something and stick it in, and it just sort of spins and grinds and does a bunch of stuff. And after a couple hours, out comes a product which they call stem cell product. It's isolated from the initial material, and there's tissue that's put into it, and that's being injected into the eye, into the joints, into the blood, and that's going on. And I know there's places here in Ottawa that are doing this, as well as Toronto and elsewhere in Canada, and certainly everywhere across the States. And the reason for this is there was no regulation for that. So because it only was manipulated for an hour or so, and it's really just your tissue that's just been modified slightly, that doesn't fall under the Health Canada regulations. It's not manipulated enough to be covered by the regulations. And so it was sort of a loophole that and still is, that doctors can use to produce a product, and of course they charge their patients for the product. Now the FDA has just begun to regulate, so they've just put in new regulations that will cover this so that they can control and shut down these kinds of clinics if they're not acting responsibly. Health Canada has not yet done that, but I imagine in the near future they will do that. So beware, if you get a call from a clinic or that offers stem cells, this is probably what they're doing.
We have another question at the back.
I just had a question regarding the stem cell infusion. So are they needing anti-rejection meds at the time of infusion or after that, or is there any concerns about allergic reactions or ill effects from getting these stem cells?
It's a very good question, especially when you're using cells that are third-party cells that aren't your own, so we call them allogeneic. But the interesting thing about these MSCs, these mesenchymal stem cells, is they don't have on their surface the usual tags that tell the body they're foreign. So they're sort of semi-protected from the immune system. So they fly a little bit under the radar. They're stealth cells, and so they don't induce an immune response. That may actually be because in the body their natural function is to induce healing, so they don't stimulate inflammation. And so that's allowed them to be used safely in this third-party manner without any immunosuppression. However, ultimately they will be removed because even though they don't have all of the usual tags that immediately tell the immune system they're foreign, they are foreign, and the immune system's pretty smart and will figure it out eventually. But they'll be removed in a very gradual way that doesn't cause any noticeable inflammation or damage. So they won't stay forever. In a way, that's a safety thing because it's very unlikely you get a tumor from these cells because the body would recognize them eventually as not being your own, and they will be removed.
So our speaker panel will stay for several minutes after the talk tonight, so that if some of you have personal questions you wanted to ask or didn't want to speak in front of the crowd, you can do that. Certainly want to thank each of you for being here tonight. It's important for us to hear from the research panel, and it's really important for us to hear from the public as well, and work together on these sorts of topics. I think Dr. Park wanted to say a few words to close our evening. No? Okay, so you're going to be free to mingle, and it's been a pleasure. Thank you.